and we mentioned enterotypes.
Okay.
Should we touch on it?
Okay, yeah, let's touch on that.
So what was or is an enterotype? Because it had a lot of fanfare for a while, didn't it?
Yes, it did. Yes, it did. Well, there was, it came out of the sequence analysis done by the MetaHit project in Europe, which is a big EU-funded project led by Dusker Ehrlich in France. So they mashed the data that they'd accumulated from large numbers of human subjects from fecal samples, and they looked at it and they decided it was not continuously distributed, that you could identify 3 clusters of microbial communities. So I think at the time, one was described as Prevotella predominant, one was Bacteroides predominant, and the third one was described as Ruminococcus predominant. Now, this was slightly muddied by the fact that nobody really knew what should be a ruminococcus in those days. It was a catch-all genus, which has now been largely reclassified. But that was. But also at the time, that it was before we'd published our paper on the dietary impact of starch and fiber. So there was this general feeling that you had a microbiome for life, and in fact, somebody
that was the wisdom.
Yeah, the received wisdom. And there was even a suggestion somebody unwisely made that these enterotypes were like blood groups.
Wow.
But they're clearly not.
That fixed.
I think the people fairly quickly backtracked from that. But so not long after that, though, some of the American groups got involved and didn't manage to find exactly the same. I mean, they did find some discontinuity, and the one that seems, but the existence of 3 groups was definitely challenged. Is it more than 3? Is it less than 3? The one that stood up and still stands up, actually, is the Prevotella bacteroides one.
Yep. Is that because of people who are vegetarian?
Well, I don't think it's that simple. I was quite mystified and skeptical about this for a long time because we'd never found many Prevotellas, but in our latest study that was published a couple of years ago in BMC Microbiology, we looked at doing a dietary intervention with 21 subjects locally here. And to our surprise, we found 8 of the subjects were Prevotella predominant, anything up to a quarter of their microbiota were Prevotella. The remaining 13 had almost no Prevotella, and were Bacteroides predominant.
And for the listener, what's special or interesting about Prevotella?
We know very little about it in the human. It's a really dominant organism in the rumen, but it's those 2 genera are both in this big gram-negative phylum of Bacteroideaceae, or Bacteroidetes, I should say. So. Superficially, they're rather similar organisms. They have a characteristic gram-negative cell wall. They produce largely propionic acid as a product, and some succinate as well. But and they have a lot of genes concerned with the breakdown of polysaccharides, carbohydrates, fiber. So you would think it wouldn't make much difference. But actually, there's something, and we still don't know what it is, that means that they don't like to coexist in the same host.
Interesting.
And they, I could come up with half a dozen possible reasons for that. Maybe we don't have time for that, but it seemed it's stood up through multiple human trials, and now that we found it in our own trial, I actually believe it.
So that's what you meant by it's kind of dead, but it's not really dead.
But it's not really dead. No, there is definitely a mutual exclusion or something that means that you don't get a lot of Bacteroides and a lot of Prevotella at the same time.
Could it be explained by? I know we're getting into the 6 things or so that you probably thought we wouldn't have time for, but could it be that, for example, the mother of the person who's Prevotella predominant was also Prevotella predominant and a vegetarian, and therefore what was seeded at birth was a little bit of Prevotella? Is that one theory?
It's one theory. Definitely. Yes. That. So there is a correlation with fiber intake, in some studies, and the classic one, actually, I think, an Italian study, but there's also an American study that shows the same thing. Is there was a group of African children with much higher fiber intake compared to a group of Italian children. African children had a lot of Prevotella, the Italian children
The Italians
didn't.
were having the Italian food.
Yeah, exactly. Lots of pasta.
Yeah,
And a similar, by Gary Wu in the States, came up with a similar thing in American populations. In our study, so we thought that was the answer too. But when we actually looked at the fiber intake of our subjects, and there was a definite overlap, complete overlap between the Bacteroides predominant and the Prevotella predominant.
Interesting.
Group. So it didn't seem to us as it was going to be as simple as that.
It's not as clear cut as
but
that.
What we did find in that was that we did an intervention with a thing called Axos, which is a prebiotic derived from fiber. So xylo oligosaccharides, basically. And that was added as a supplement in this study. And what we did find was that Prevotella was greatly encouraged by the Axos.
And is that a good thing?
That's the jury's out on that.
So should everybody have a fiber supplement if they're not getting enough through their diet?
With that, you're taking me in a different direction now. But it depends where they sit on the recommended intake scale. So they should try to achieve the recommended intake. Whether they do it with a particular prebiotic, many people would say that a complex fiber is better, in general terms, than a specific supplement.